The Alaska Marine Science Symposium is a premier gathering of researchers, scientists, and resource managers in Alaska every year to discuss the latest in coastal and marine research and findings. This year, one of the keynote speakers was Jeff Short, an Exxon Valdez oil spill scientist, and he was sharing with us information about the scientific legacy of the Exxon Valdez oil spill. This is the 30th anniversary of the spill this year, so it was one of the themes at the symposium this year. In his talk, he emphasized that the Exxon Valdez oil spill was a once-in-a-lifetime opportunity due to the way circumstances came together, and I'll describe each of these bullet points in a little more detail in a moment. He also said that we really saw a switch to ecosystem-level studies as a result of the Exxon Valdez oil spill, and that the spill really did have a lasting legacy in Alaska in the way we do science on the Alaska coast today. So regarding this once-in-a-lifetime opportunity, he said, this was really one of the most ecologically damaging oil spills it was known of, and that's for a few reasons. For one thing, it occurred during oceanographic springtime. This is a time when plankton are blooming in the Gulf of Alaska, and therefore you have a lot of migratory species that are headed up here to take advantage of that plankton bloom and of that abundance. But they were concentrated on the water, so some of the species that might end up on land, like seabirds, weren't quite on the nests yet, but they were all concentrated on the water where the spill took place. So, you have this highly productive ecosystem. It attracts a high biomass, and that biomass was right in the pathway of the spill. You also had a relatively pristine marine ecosystem, and we think of Alaska as pristine partly because of the sparse human population, but there was also a unique set of circumstances in Prince William Sound where you had a relatively new intertidal zone. So in the 1964 quake, a lot of the intertidal zone was sort of reset. Areas of land rose on one side of the epicenter, areas of land dropped on the other side of the epicenter, meaning that what was once intertidal came up out of the water in some cases, and those things died, or other things were sent deeper. So you had this new intertidal that had been developing over the last 20 years or so since the quake, and being so new, it had little exposure to environmental degradation. And so he really emphasized that this was a relatively rare set of circumstances that provided an opportunity to study how oil affects natural ecosystems. So one of the things he said we also saw from the spill was a switch to ecosystem level studies. And, in many cases there's kind of a tension between scientists, attorneys, and the public desire for knowledge. And I don't mean in many cases, really in this specific case there was that tension. And what scientists wanted was knowledge, what happened out there and what are the effects of what happened. The attorneys, on the other hand, were in a situation of wanting to build a case for litigation, and so a lot of their information that they wanted to gather needed to be kind of secret. They weren't sharing a lot of it. The public, of course, just wants answers. They want some simple answers as to what's going on. And there's a tension between all these three desires. And one of the first sort of opportunities that scientists took after the spill to kind of try to share this information with each other and maybe respond to some of that tension, get the information out there, was this big symposium in January 1993 in Anchorage. They wanted to get together to share the current state of knowledge. And really that gathering was the precursor to what has now become the Alaska Marine Science Symposium, which happens every year in January in Anchorage. And it was at that symposium in 93 that Jeff Short feels that there was really a realization among the participants that broad ecosystem level studies were warranted, were required, were what was desired to understand the effects of something like the oil spill. You couldn't just study a single species. You couldn't just be a sea otter biologist and say, I'm just going to focus on sea otters alone. You really needed a lot of information from other scientists about other aspects of the ecosystem to understand what was happening to sea otters, for example. So this switch to ecosystem level studies grew out of that first symposium when finally in 1994, based on recommendations from the symposium, there were a first commitment to multi-year interdisciplinary and multi-agency studies. And those studies took a few forms, or a few names actually. There's the Sound Ecosystem Assessment, the Nearshore Vertebrate Predator Project, and the Alaska Predator Ecosystem Experiment that all came together in the mid-90s there. And these are all things that you can look up and see the legacy, the reports from these studies. They're still out there and became sort of a model for these ecosystem level studies. Since the spill, there's been approximately $250 million spent on science. And some of the results are that scientists learned that the ecotoxicology of oil was quite different than anticipated, that the oil persisted quite a lot longer than they anticipated, that there was an ongoing toxicity of oil 30 years later. They thought that was surprising. A lot of people thought, volatiles and so forth would evaporate and the oil would wash away and there wouldn't be this lingering toxicology that we do have out there. And then one of the other important results is the development of these long-term databases. There was an increase in the number and the quality of data in long-term databases, particularly in relation to orcas, sea otters, the Seward line, which is a set of oceanographic data that's taken on a line that goes straight out from Seward, out Resurrection Bay, out to the continental shelf. And then sort of the ongoing legacy of these long-term databases is the development of Gulf Watch, which is continuing to add data to databases on both the pelagic ecosystem and the nearshore ecosystem throughout the spill-affected area. And then finally, he talked about the lasting legacy of the oil spill and, emphasized that the Alaska Marine Science Symposium is linked directly to that first EVO symposium in 1993. And that most importantly, the legacy is how it's done, it being science, the way we do science in coastal Alaska now. There's a competitive review of proposals. There's inclusion of the private sector and universities in work that agencies are conducting or are interested in. There's been a breakdown of agency territorialism. We're not as stovepiped anymore, and we see a lot of projects that include multiple agencies. Talked about an adaptive management cycle, which is sort of looking at results along the way and changing management practices as we go. Peer review of science and stuff that's done in relation to the spill-affected area is common now, and it seems like maybe a no-brainer, but honestly, when I first started working in the Park Service 20-something years ago, a lot of park science was not peer-reviewed. It was published in white papers internally or published in our own park science publication, but not a lot broader than that. So that's changed, though. That's definitely changed over my career. We see ecosystem level studies. I think we've talked about that enough. And we see this annual symposium for the sharing of information. All these things are legacies of the Exxon Valdez oil spill. So that was a rather detailed look at his talk. For the next talks, I'm going to have one or maybe two slides and just hit the main points. So there was a talk given on the impact. So these next few relate to ocean acidification, which is a big issue that we know is happening out there. So this one is about elevated variable pH on the Pacific razor clam in Alaska. This was done by a master's student, and there's an interview that's linked at the bottom of this, which of course you can't hit the link during the video, but you can find an interview with Marina Washburn with a simple Google search that goes into some detail about this work as well. But she did an experiment at the Aleutic Pride shellfish hatchery in Seward. She exposed embryos to both elevated and variable levels of CO2. So we know that levels of CO2 are rising in the ocean, but this isn't a constant. It's something that varies. And she did this variable level to simulate the photosynthetic drawdown of pH by photoplankton. So pH varies over the course of a day and night cycle depending on how much photosynthesis is happening out there. So as it gets darker in the evening, less photosynthesis is going to change the balance of CO2 and pH in the water. So what she found was that shells in these Pacific razor clams were shorter by day 28 than in animals that were exposed to the elevated pH levels. But she also found out that the elemental composition of the shell was different from animals that weren't exposed to the pH. And this suggests to her that there's more resilience than anticipated. In other words, what it means is they're able to switch the composition of their shell to something else that may be more readily available out there to them than the calcium carbonate that is predominant in most shells. So this comes from something that she calls a concretion that starts at the very beginning of shell development in razor clams. So they start their shell with this concretion of many different elements. And the fact that they do this may mean that they're a little more resilient to ocean acidification than we thought they might be, because it seems to give them the ability to sort of switch the composition of their shell to other elements in the case that something else is not as available. Now, the interesting thing is this concretion is not common among all clam species, and even among other animals that are create shells as well. So we don't know how widely spread that is, that concretion, but it's not the norm. Not all shellfish do that. So that was sort of an interesting finding there. Okay, the next one was a comparison between ocean acidification in high latitude. So she compared three different geographic seas or areas: the Gulf of Alaska, the Bering Sea, and the Beaufort Sea. Of course, we know it's all connected, but these are somewhat isolated bodies of water, even though they are ultimately connected as all part of one ocean system. But so what she did was look at pteropods. And pteropods, some of you may know, are like these, they're sometimes called the butterflies of the sea. They're these little tiny snails that have their soft parts flapping in the water, kind of like butterfly wings. You have to look up a picture. I should have put one on here. But suffice to say, they're sometimes called butterflies of the sea. They're little tiny snails, but they're very important because they're a major food source for juvenile fishes in the sea. So they're this important link in transferring energy to higher trophic levels. They don't have concretions in the larval stage, so they don't make a concretion of lots of elements. They're going straight to that calcium carbonate shell. And there was not much genetic variability in them, suggesting that they would have a low adaptability to ocean acidification. Not a lot of mutations going on in the not a wide variety of mutations in their genetic code, so you would think that means less adaptability. What she found was in the Gulf of Alaska, there's already undersaturation, meaning conditions where shell building is challenging for pteropods because there's not as much of the calcium carbonate as they would like to find. It's already at detrimental levels in the spring, and we see some shell dissolution already occurring, and this is worse in the western Gulf of Alaska. In the Bering Sea, we see undersaturation almost year-round, and we see some shell dissolution showing up on the inner shelf. And finally, in the Beaufort Sea, there is prolonged undersaturation, particularly in the fjords systems up there. And her sampling there showed 70% of the shells observed were already showing dissolution. So a challenge or lack of ability to create a robust shell for those pteropods. So bad in the Gulf of Alaska, a little bit worse in some places in the Bering Sea, and the worst effects that she observed were in the Beaufort Sea. So another talk looking at shellfish was Mike Booz from ADF&G giving a little perspective on the recovery of razor clams in East Cook Inlet. And this is East Cook Inlet is, of course, where there's a sport clamming along road system there where people would go into Clam Gulch and the Nilchik and so forth to harvest clams. Traditionally, that has been closed for a while. So he's given a little update on what ADF&G thinks is going on with the razor clams there. Not super conclusive, but here's some of the data. He started off by saying in the 13 years he's been at ADF&G, he has seen declines in virtually all major sport fish stocks. So that was a real, realistic, I suppose, but somewhat depressing little fact to start off with. He said razor clams are no exception. There have been big declines that he's seen, and it's been closed in Eastern Cook Inlet since the beginning in 2014. What they are seeing now is an increased density in juveniles, and they've observed that already by 2016, and that increased density of juveniles continues, but there's adult density is still low. So based on what they're seeing now, the fishery will remain closed through 2019. He's cautiously optimistic about recovery, but sort of the mystery is how come they're growing to juvenile age, but they're not making it to that adult stage? What's happening in making that transition from juvenile to adult? We don't really know. Another talk that was given, this is moving away from shellfish, is looking at the success between hatchery strays and wild pink salmon in a natural stream system. And so, there's a lot of hatchery fish that are released into Prince William Sound and the Gulf of Alaska and other parts of Alaska. And the question is, are these hatchery fish wandering, some of them up into streams and, diluting the wild stock with their hatchery genetics? What Kyle found was that, let's see. Okay. In most species that have been studied in the past, we do see a lower reproductive success among hatchery strays. Just hadn't been studied in any. That was his study. This was one looking at, and we're going from the little stuff in the sea to the big things, looking at humpback whales down in Southeast. And Madison Cosma worked at a hatchery, which gave her a unique opportunity to observe whales in a unique setting, because the hatcheries, these whales are hanging out to feed on hatchery released salmon. It's kind of a new phenomenon. Whales are smart animals. They've figured out that there's these regular releases of tons of little tiny fish, and that's one thing they like to feed on. They'll school around herring, around sand lance, around small fishes, and feed on them. So they're learning to hang out around these hatcheries and wait for releases. So it provided a unique opportunity because there's a series of docks and stuff that she could go out on and get very close to the whales. She also had a nice platform there on the docks to launch drones from, so they could fly above the whales and get a perspective there on what the whales were doing. And what she observed, she calls behavioral plasticity. I don't think that's her term. I think it's a term that biologists used to describe new, unique, and adaptive behaviors. The animal will take a behavior they already have, they'll encounter a unique situation, and they'll modify that behavior to match that situation. So her behavior that she saw, she calls pectoral herding. And there were two types of pectoral herding documented. I have a couple pictures here in a second that show what she observed. Whales using their pectoral flippers to help herd fish in a way that hadn't been documented before. And she thinks one reason they may not have been doing this before was that juvenile salmon are a unique prey item for whales, and these concentrations of juveniles right outside a hatchery may be unique as well. Like they kind of disperse once they get out in the wider sea, but they're being released in these masses. So even though it's a tight school, it's a little more loosely aggregated than some schooling species like herring that they focus on. And so the techniques they use for herring may not be quite as effective on salmon, so they're adapting their techniques to make them more effective for this prey species. 1, 2, flipping, 3, then feeding on the fish that have been herded in that way. The other type was this pectoral herding, where they're coming up through the bubble net, but then using the pectoral flippers to push the fish toward their mouth with the flippers. But she's theorizing that not only are they pushing them and actually moving the water with their flippers, but they're actually pretty interesting study. Interpreters who are out there talking about whales and the various ways they feed will find this interesting to add to their description of whale behavior. Speaking of whales a little further, Jan Straley from Southeast Alaska gave a talk about the decline in reproductive success that is happening with humpback whales in the Gulf of Alaska. She started with the good news, which is that humpbacks have been on a strong recovery path over the last 2 to 3 decades, really. And the Hawaii population, which is the population that primarily comes to Southeast, most of Southeast Alaska whales go to Hawaii and then they come back in the summer to Southeast. That population segment was delisted from the endangered species list. They're not considered endangered or threatened anymore. I mean, they're doing really well. But in the past couple of years, very few calves have been observed. And when a species is recovering, it's eventually going to reach something that biologists would call a carrying capacity. So ask the question, is this an indication that carrying capacity has been reached? That they're not producing as many calves but she threw that question out there and then kind of rebutted it herself by saying it doesn't appear that this is just happening to humpback whales. It appears that multiple species are affected in the past couple of years, suggesting that while humpbacks may have been headed towards carrying capacity, they may now be caught up in a larger ecosystem issue that's having a broad effect. Seabird die-offs, other marine mammals dying, in large numbers, sea lions in California, some other whale species in Alaska, seabirds around the Gulf of Alaska. And she believes this is probably an indication of a decline in prey availability rather than something indicating a carrying capacity. So nice recovery story for humpbacks, but sort of a caution. I have just a couple more talks to tell you about. This one is looking at the Bering Sea cold pool. And if you haven't heard of this, there's a pool of cold water that forms and stays year-round in the Bering Sea. And in the summertime, this cold pool becomes what they call a thermal curtain. I suppose that curtain is there year-round, but it's most important in the summer when things are breeding, reproducing, moving about. There's high productivity in the Bering Sea. But what this the cold pool does is form a thermal curtain, and certain species that don't like that water that cold stay south of that thermal curtain, and other species are north of that curtain and up into the Bering Strait region. And that cold pool has been declining in recent years, and in fact this last year was the first year that no thermal curtain was detected at all in the Bering Sea, and biologists saw some anomalous observations of where species were found and what species were found in what abundance in the Bering Sea. I won't go into details of that. Look it up if you want. Bering Sea thermal curtain or Bering Sea cold pool. But in a specific study, she was looking at snow and tanner crab in the Bering Sea. So there's been a historical decline in both species in the Bering Sea. We know that in cold water, you have higher fat zooplankton. So the feed, the basis of the food web, is more nutritious, higher in fat in cold water. In warmer water, the metabolic rate of zooplankton increases, meaning they grow faster, so they may be bigger, but they have less fat, which makes them a less nutritious food in some ways for fish, birds, mammals that may be feeding on zooplankton. So her lab experiments and field observations support this. So she did some lab experiments to grow zooplankton and sort of supported what we already kind of knew was true about cold water and warm water zooplankton. She was able to observed that the geographic range of snow crab was contracting to the north in relation to the contraction of the cold pool. So the geographic ranges of animals are changing as the cold pool changes. Not a real shocker, or not a real surprise, but we're going to see a lot of that kind of thing happening as ice disappears, as cold water changes to warmer water. And then finally, I want to tell you a little bit about an experiment that was done at the Alaska Sea Life Center looking at threatened Steller's eider and spectacle eider ducklings. These are two species that do winter along the coast of Katmai National Park, and then over on the other side of the Alaska Peninsula as well, in the Bering Sea. They do a lot of their nesting on the Yukon-Kuskokwim Delta. And so sea level rise, lack of sea ice, and increased storm activity are pushing salt water into the low-lying Yukon-Kuskokwim Delta breeding area. That's what YK breeding area means, Yukon-Kuskokwim Delta. So there's a lot more saltwater inundation in there. Newly hatched eider ducklings do not have a fully functioning salt gland. So these birds, they live on salt water as adults, and they're constantly taking in salt water, so they have a salt gland that helps get the salt out and make that water useful to them, not harmful to them. So what they did was take some newly hatched ducklings and they exposed them to salt water that simulated levels detected in the areas of the Yukon-Kuskokwim Delta. And what they found was that if they had 3 parts per thousand salinity, there were no effects on the ducklings. But if they went up to 6 parts per thousand, they saw clinical and behavioral pathology where the ducklings were pretty severely impaired. They couldn't walk right. They almost appeared drunken. They couldn't balance themselves. As soon as they detected these pathologies, they stopped the administration of the salt water and gave them fresh water. So they assured us that no ducks were lost or permanently harmed, in conducting this experiment. They also found that if they delayed the exposure to salt water until day 8, the 6 parts per thousand had no effect. So really, these ducklings, they do develop a salt gland that can deal with this. It's just those first few days out of the egg, they need access to fresh water. And this increasing inundation of their breeding grounds with salt water could prove to be a critical or limiting factor in their recovery. All right, I'm going to turn it over to Deanna Ux to tell you about a few of the talks that she attended at the symposium.
For the first study I'd like to talk about, it has to do with the relationship between brown bears and sea otters in offshore islands of Katmai National Park. Researchers wanted to know if brown bears were preying on adult sea otters. They're not known for hunting adult marine mammals in particular, but they were finding these sea otter carcasses in these offshore islands that had apparently died during the prime of their life, and they wanted to know why. So they trained cameras on sea otter haul-out sites to try and figure out what was happening. So what they found was indeed bears were stalking and preying on the adult sea otters. And this was very unexpected, unusual behavior because bears were known to be more opportunistic feeders. They would feed on carcasses, but of animals that had already died. But these were sea otters that were dying in the prime of their life. So what did this mean? That sea otters are apparently, at least in these cases, important food sources for some bears. However, because sea otter populations are stable or growing, it doesn't seem that this is real typical behavior. What it is just a little known interaction between two predators, and it may have an impact on the intertidal communities of south central Alaska. The next study I would like to talk about is a study of the abundant or the ongoing seabird mortality events that have been occurring in the Bering Sea. Researchers wanted to know if there was a link between these events and a warming ocean. What they'd noticed is that their bird die-offs have occurred annually since 2013, and previously they only occurred every 5 to 10 years. They also note that these events have coincided with warmer ocean temperatures and reduced sea ice. To conduct the study, they gathered 5 years worth of data on the die-offs in the Bering Sea, and then they compared them to similar events in the North Pacific, as well as historic records. They found that the number of birds affected is increasing, the types of birds affected has been increasing to a broader range of species, that the birds' ranges have shifted north, and that their prey species has also shifted. And they've believe that these mortality events are going to become more frequent as the climate continues to become more and more variable. The next slide, the next study I'd like to talk about has to do with Chinook salmon and why they seem to be coming back to spawn, why they're smaller when they return to spawn than they have been in the past. Typically spawn return at age 4, but in recent years they've begun returning at between ages 2 and 3 more commonly. And when this occurred in Canada, the fishery collapsed. So they conducted the study by tagging salmon to track their movements and compared these findings to previous population tracking models. They found that indeed age 3 Chinook salmon were dying more frequently, up to 46% more frequently. They were finding that the salmon were being preyed on at age 3 by salmon sharks and killer whales. And the interesting thing about this is that typically it had been believed that salmon after their first year of life were fairly safe in the ocean, but this would indicate that that is not the case. The next study I'd like to talk about has to do with an invasive tuna called Duodenum vexillum. Duodenum vexillum has been found recently in Sitka, Alaska. And the researchers wanted to know if it was possible to eradicate this tuna also called rock vomit. It was first found in Sitka's Whiting Harbor in 2010, having come north from potentially as far south as Japan. And since 2010, it has actually gotten to the point where it covers parts of the floor of the Whiting Harbor and smothering organisms in the process. And it is currently threatening to spread out from the harbor, and there is currently no known way to get rid of it. To conduct the study, they put turbidity curtains in place to isolate the area, and then treated them with lime, salt, and chlorine. And they found that while the lime and the salt were not successful or very minimally successful, chlorine was actually quite effective, and it was very easy to use. It was very practical, and it was effective in removing most of the Duodenum vexillum in the controlled areas. So the study suggests that chlorine may indeed make it possible to eradicate this tuna that had been previously thought to be almost impossible to get rid of. This study has to do with the what an ice-free winter might mean for the Bering Sea. Researchers wanting to know how an ice-free sea might affect the ecosystem. 2017 and 18 had the lowest record of sea ice since 1978 on the Bering Sea. And the sea ice provides important habitat for algae and plankton. So a reduction in the ice also means a reduction in high energy food for zooplankton. They conducted surveys of the oceanic conditions in April and May of 2018 and discovered that zooplankton, in particular copepods, were abundant, but they were smaller and they were lower in fat. The spring bloom of phytoplankton was delayed by a month, and small fish such as pollock were moving northward in pursuit of food. It had the smallest cold pool on record, and animals were adapting. For example, fur seals that they had tracked had changed their diet from pollock to salmon, and this required much longer diving times and greater expenditures of energy for the fur seals in order to capture their prey. The next study also deals with sea ice conditions, and this is on the Chukchi Sea, and researchers here wanted to know why the sea ice levels in 2018 were so low. What was the cause of it? 2017 brought the lowest ice extent on the Chukchi in 40 years, and the ice that did form was very thin and very mobile. So they reviewed data sets and satellite observations of the different factors driving the ice extent, and they found that much warmer air temperatures, even above freezing in the winter. They found that ice was forming 30 days later than it had between 1981 and 2016. That this was resulting in warmer temperatures on the ocean's bottom and had reduced the cold pool dramatically. And most importantly, they discovered that the wind direction had changed, whereas they had previously come from the north. Wind direction had shifted and it was coming from the south, which then delayed the arrival of ice and brought warm water in from the south. And the predictions in the future are for this trend to continue, which will set the stage for more low ice years and, again, small cold pools.
This next talk was given by Dr. Katrina Cunahan from the Alaska Sea Life Center. And actually, this work had some National Park Service staff involved as well, myself, Benjamin Pister, and Heather Coletti from the Southwest Alaska Inventory Monitoring Network, as well as some of our colleagues from USGS and the Alaska Sea Life Center. The title, Physiological and Gene Transcription Assays in Combination, a New Paradigm for Marine Intertidal Assessment. That's kind of a mouthful, so let me give you a little bit of background. Typically, our monitoring programs look for changes in a species population. Is the population increasing or decreasing? Or we may monitor a physical variable like temperature. And then when we see changes in those variables, we look for reasons why, either in the data we have, or maybe we design a new study to help us figure it out. And we know there's a lot of big changes happening on the coast, and a lot of those things are either expensive to monitor because they're so remote, or it's simply difficult to do. So we're looking for a way to use organisms themselves to give us a sense of what they're experiencing. So the approach we took here, which is led by Dr. Cunahan, looks at mussels found in the Rocky Intertidal. So those mussels are found in a variety of parks, and they're eaten by a lot of other animals. And we tried to combine two different approaches. One was to measure physiological characteristics, things like the ratio of the weight of their soft parts to the weight of their shell, or how much hydrogen peroxide they produced, which is something mussels use as an immune response. The second approach was to look at their gene expression, or transcription. In a nutshell, the genes encoded in DNA are used to make proteins. When an organism needs more of a particular protein, say a heat shock protein to protect against thermal stress, it uses the gene more often to make that protein. Now, generally the amount of DNA in a cell stays the same, But when a gene is being used more, that is, more protein is needed, the amount of RNA transcribing that DNA into proteins goes up, and we can measure that RNA. So if we know which genes code for heat shock protein 70, and a muscle uses heat shock protein 70 when it gets too warm, then we can measure the amount of transcription RNA for heat shock protein 70 in a muscle, and we can tell whether it's been experiencing temperatures that are causing it stress or not. There's some moderately complicated biology underlining this process, so if it doesn't make sense or you want to know more, give us a call, or you can Google it or look in a textbook. So what did we find? Well, it's still a bit of a work in progress, but we identified 7 of these physiological characteristics that we would call them biomarkers, and 14 genes in the gene expression part. And one of our goals was to simply establish a baseline for what it looked like in these organisms, these mussels, in relatively pristine areas. So we did that for mussels in Lake Clark and Katmai in 2015 and 2016. So that's something we can use to compare to later changes. But we also kind of couldn't help look for differences. And there were some between Lake Clark and Katmai. The Lake Clark mussels had thicker shells and more soft tissue. That could mean they had better nutrients, more food than the Katmai mussels. And the Katmai mussels also showed a greater immune response. So something was making them not feel quite as well. It doesn't mean they were sick or diseased necessarily, but their immune systems were working a little harder than the ones in Lake Clark. What this approach could do is let us understand what organisms are experiencing and what they're not. It may allow us to do a lot less work in the field for a lot less money and discover a lot more about what's going on. For example, we could potentially use this approach to monitor for oil contamination, diseases, meaningful changes in temperature and pH, food availability, and several other factors. Still a work in progress. There's a lot more lab work to be done, but we're hopeful it will increase our tools, the tools available to monitor the environment in our parks. We're also doing this work on razor clams from Katmai and Lake Clark, and for anyone who's familiar with the Changing Tides project, this is a study that is part of that larger project. Tune in next year for the rest of our results from razor clams. So in addition to the studies we've talked about so far, there were several talks at AMSS that may be of general interest to a lot of National Park Service folks, but may not have had a very strong connection to the national parks in Alaska. Rather than dive into each one of them, many of these can be grouped into general themes. The goal here is to give a general sense that this work is being conducted, and maybe it gives you some ideas for your own work or for work with a new partner. If it does, you can track down these studies in the online abstracts or ask us and we can help connect you to them. The first theme I'll talk about was use of autonomous vehicles. So we're used to thinking about unmanned aerial systems or aerial drones in the National Park Service as a new tool, but there's a lot of different autonomous vehicles or drones, if you will, being used all over the place. At sea or in the air or underwater. Principally, these systems do one or more of 3 things. 1, they go where it's hard for people to get to. 2, they stay somewhere longer and cheaper than people can. Or 3, they disturb the environment less than if people went there and did the same thing. So there were at least 5 different talks that were presented using autonomous vehicles of some kind. So Madison Cosmo used a quadcopter to observe humpback behavior, and Jim talked about this in another slide, so I'm not going to go into detail about it, but the point was Ms. Cosmo used a quadcopter to observe whale behavior from a perspective that she couldn't get otherwise. In this case, it was just above her and nearby, but it was critical to understanding what was going on with the whales. Another one I'll mention is the sail drone. That's that red sailboat looking thing on the right. It's a little bigger than it might seem in the picture. It's 7 meters long and 3 meters high or more, and it is essentially an unmanned sailboat. It's powered by the wind. In terms of propulsion, and the systems on board are solar powered, and it can be controlled by satellite link. And it is deployed from a harbor or from a boat, and it can stay out for months at a time. And this year at AMSS, there can be a lot of different packages on board, a lot of different instruments. But this year, McCurran et al. Discussed how they used it to map pollock preyed on by northern fur seals in a multi-month deployment. So it sailed around using sonar to count the pollock in the areas that fur seals were feeding, and they were able to get quite a lot of data without having to go there themselves on a much more expensive research vessel, and they could track that prey over time. Another kind of interesting UAS vehicle is called the EB, which is the black and white plane looking thing in the lower right corner. So this is a UAS, and this was used by in the Pribilofs. They used a couple of these things at the same time. So in this study, the municipality of St. Paul actually used multiple EBs flying in tandem to map or to actually count harbor seals. And these things had a good 45 minutes of flight time. They had one flying right behind and above the other. And one collected visual imagery, visual pictures, and the other one was collecting infrared imagery. And when you process them together, they could be used to count pretty accurately the number of harbor seals, which the community uses for subsistence. So that was a citizen science sort of driven use of autonomous vehicles. There's another one called the Oculus. There's a picture of it down at the bottom. This is these are basically little submarines that are deployed by NOAA, and they can be used in areas that are kind of shallow for big vessels to go, but they can also be deployed for quite a long time to collect oceanographic data. There's some other kind of cool ones that are used to study under ice environments, reported on by Phyllis Stabenow and her team. And basically they could deploy these things in the summertime on the bottom. They stay moored on the bottom until the ocean freezes, and then a timer releases the floats on their way up to the surface. They collect all kinds of information on the water column, and then they get jammed up under the ice, stay there until the ice melts, recording data the entire time. And then when the ice melts, they pop up to the surface and beam their data home. So who knows, kind of a creative application, maybe something someone could apply to some of the lakes or in our parks. So the other theme I want to talk about, and this will be the last slide for me, is the use of collections. So if you're ever wondering why collections are important, or besides simply preserving artifacts, there's actually quite a fantastic amount of data that can be gleaned from various collections, and there were several talks that were taking advantage of that this year at AMSS. So here's just a few of them. So one study was using isotopes of amino acids in sea lion whiskers from different collections to study seasonal changes in the food web. Another study by Curel et al. Was doing something similar with northern fur seal teeth. So these were teeth that were taken out of museum collections, and they were able to reconstruct entire food webs from the Bering Sea from 1946 to 2014. So the cool thing about this is they're able to go back and ask ecological questions from time periods when field scientists were not out there collecting any data. A really cool study by Caitlin Meadows used museum lots of bivalves, that's clams, from the Bering Sea. And if you're ever looking for a reason to collect good metadata, look at this study. But Ms. Meadows was able to use the metadata from the labels as well as the shells themselves, and intake ledgers from the museums, coupled with cruise reports from the ships that were used to conduct the work back in the day, and personal journals from people on those boats to reconstruct what the benthic community, that's what's growing on the bottom of the Chukchi and Bering Seas, look like over the past 150 years. And one of the key questions that Ms. Meadows was asking about was, we started collecting ecological data back in the early 1970s, and we've kind of wondered, was that how it always looked? Because it's changing a lot since then, but did it look like that earlier, or was 1970 just a snapshot in time in a changing environment? It turns out it's the latter, and that bivalve community in the Chukchi and Bering Seas in the early parts of the 20th century didn't look anything like what it did in the 1970s. It was a little bit more similar to what it's looking like now. So gives us a much more complete understanding of what's going on from time periods when we didn't have biologists out there collecting data. Another study is looking at bird feathers, black-legged kittiwakes, and some other species to look at shifts in their diet and the timing of their migration, and in a really cool way, the timing of the sea freezing. So basically, by looking at these feathers in collections, they're able to infer when sea ice covered the area. And the kind of cool thing about this is these species are found all over the north, so they can compare what was going on in Alaska to what was going on in Greenland or other places, and they can also look in the past and figure out what was going on with the ice and when they were migrating and what they were eating. The last one I was going to mention was using walrus teeth. So Clark et al. Measured zinc concentrations in the tooth cementum. Cementum's laid down daily, so there's a long record of it throughout the life of a walrus. And the amount of zinc, or the cementum, can be used to determine gender and age, and the zinc concentrations can be used to determine how old that walrus was when it was sexually mature. And from that, you can infer population size, and from both of those, you can infer what the resource availability was for the entire population. There's over 10,000 teeth in museums in Alaska alone, and every walrus that's taken now has a tooth extracted to determine age. So there's a very long, full record of walrus teeth in the museums, and you can answer some fascinating questions about what was going on in the environment without actually having to have had people there collecting the data in person. So museum collections are a powerful tool.